Alright, hi, my name is Casey Cooper, and this is my presentation of Minamata disease, gene editing technologies, and unforeseen consequences. This will explore the Minamata disease and the motives behind it through the lens of Japanese culture at the time, and relate its consequences to gene editing technology and its role in the far future using deterministic concepts. Now, what we have to establish first is the general link between Minamata disease and gene editing technologies. The core linkage between them is the idea of multi-generational unforeseen consequences. Unforeseen, not only because of the indeterminate nature of genomes and the time it takes for latent effect, but because there is a strong presence of plausible deniability against such unintended effects. These effects are often slow and delayed, as well as incredibly multifaceted in their behavior, making them difficult to predict or gauge to the average person, as they may only appear in someone's children or their children's children, and so on. I will go further into this later, but we have to get some background first. The Chiso factory in Minamata, Japan, was initially constructed to produce fertilizer. However, they eventually became a major producer of chemical substances, one in particular being acetaldehyde, synthesized using mercury sulfate. The factory frequently dumped their wastewater into Minamata Bay, a crucial food source for the surrounding area as well as a job supplier for fishermen. Despite the fact that they had already faced environmental compensation fines to the local fisheries for minor damage, they did not hesitate to continue even after acetaldehyde production, starting in 1932. You can imagine that dumping fertilizer-related wastewater wouldn't necessarily harm the environment nearly as much as dumping mercury. However, this didn't seem to cross their minds. In 1958, the first case of Minamata disease was reported among many others soon later. It was dubbed an epidemic of an unknown disease of the central nervous system. This is crucial to the idea of plausible deniability, as due to the lack of information, this led some to claim that it was contagious because of the close proximity between cases, among many other distorted claims. It took 2 years before they traced the source to metal, and consequently the factory, and then 1 more year to figure out it was mercury, as it was difficult to differentiate between the various other possibly toxic metals dumped by the factory. So, what was the Chiso Corporation doing at this point? Well, they essentially didn't really care for the damage, and they went through much effort to absolve themselves of any blame. First, upon facing general scrutiny for the disease, they started off by redirecting the wastewater route from the original harbor directly into the Minamata Bay. Almost immediately, dead fish began washing up on the beach, and many more cases of Minamata disease were reported in other fishing villages. Once they were contacted by investigators, they intentionally misguided and withheld information from them. When a Chiso researcher began conducting experiments within the factory to really see how much the mercury was affecting the environment and its inhabitants, it was tested on cats. The results were astoundingly concrete that Chiso was poisoning the population with mercury. After this discovery, Chiso did not allow these results to be released and made their researchers stop immediately. But it still doesn't end there. Now that Chiso were certain they themselves were responsible, they hired other researchers in hopes to mislead the public and generate an alternative source of the cause, unrelated to their wastewater. This is the plausible deniability factor that they tried clinging on to, as the mercury discovery was still relatively recent. The Chiso Corporation's efforts were not enough to redirect blame, and all attempts to come to terms with their actions were usually compensated for with sympathy money. And that's what they called it, but the etymology could just be different in Japanese, but it's kind of a cruel term if you think about it. Alright, so now we're gonna, analyze the actions of the Chiso Corporation in the context of the NSP Code of Ethics. and we find that they violated at least 5 of the rules, and we're going to run through them now. So the first, obviously, being to hold paramount the safety, health, and welfare of the public. So the Chiso Corporation knowingly discharged methyl mercury into Minamata Bay for years, even after evidence showed it was harming local populations. The Japanese government failed to act decisively or transparently. Chiso engineers and executives should have ceased discharges immediately upon signs of harm. The government should have prioritized public health investigations, enforced industrial accountability, and provided clean alternatives to affected communities. Transparency should have been non-negotiable, not protecting corporate interests over human lives. So the second one that Chiso violated being performed services only in areas of their competence. Chiso company doctors and internal researchers attempted to cover up or mislead the public about the true cause of the disease, essentially manipulating science to fit a desired outcome. Independent, multidisciplinary scientists should have been brought in immediately. The government should have empowered unbiased environmental and medical experts to assess the situation, not allowed company-funded pseudoscience to dominate public narratives. The third rule that Chiso violated was to issue public statements only in an objective and truthful manner. So, public messaging from both Chiso and government officials was often downplayed, distorted, or just denied outright. Victims were even stigmatized or accused of fabricating symptoms. We can see that in the fact that they thought it was contagious, so they ostracized other people. And the government should have honestly communicated health risks, even at economic cost. Engineers and public health officials had an ethical duty to warn the public clearly and act preventatively, even without full scientific certainty. The fourth rule that the Chiso Corporation violated was to act for each employer or client as faithful agents or trustees. So the employees at Chiso in the Minamata factory, they all had an option to advocate for the correction of the waste they were putting out, as well as treatment of all the affected people. But almost all of them complied with Chiso and made efforts to suppress information and not really advocate for anything or have any whistleblowing action going on. And so, ethical engineers should blow the whistle if employer conduct endangers the public. The company should have welcomed transparency and correction, not suppressed data or silenced internal critics. And so, the fifth and final one that Chiso violated was to avoid deceptive acts, and that one was pretty established before. Chiso manipulated environmental data, denied responsibility for health damages, and paid off local officials. The government also delayed official recognition of the cause of the disease for years. All data about mercury contamination and health effects should have been published and peer-reviewed. Any engineering work connected to industrial discharge should have included environmental risk assessments and public disclosure. Alright, so now we're going to get into a little more in-depth analysis behind the reasoning of the Chiso Corporation and the Japanese government. So, to me, this seems like a case of post-war Japanese utilitarianism, which is influenced by their historical and cultural emphasis on honor, the highest of Japanese virtues. The rapid industrialization of Japan was brought about from a strong desire for national recovery and pride after an understandably crushing war effort, leaving most individual concerns behind for the sake of redemption, where honor and self-annihilation are one and the same. We see this historically in the samurai and their art of seppuku, in kamikaze pilots as a last resort, as well as Japan's stubborn presence in the war, even after it was largely considered over, out of fear of a humiliating surrender and strong adherence to the Bushido Code, commanding honor and death over surrendering while their citizens get irradiated en masse. The Chiso Corporation, in particular the Minamata factory, did in fact play a crucial role in the development of Japan's rising chemical industry, and was also a large source of commerce, making these motivations a likely factor in the decision-making of those in power. The deliberate lack of sufficient action taken, which lasted 12 years, makes it seem that to Chiso and the rest of Japan, Minamata disease was a rather fair price to pay for Japan's glorious industrialization, just as a young pilot's life in an airplane seemed a good enough trade-off for a sunken American battleship. This is absolute, pure utilitarian beliefs, and Japan was not going to let a couple fishing villages stop their expansion. Despite these dogmatic cultural factors, I believe there is also a distance factor to it. If the executives at Chiso had perhaps been shown, in person, the morbid effects that were afflicting the citizens, they could possibly have second thoughts. Though honestly, I'm having my doubts about that. Although, it is much easier to be enthusiastic about a war when you aren't faced with the horrors of war, and likewise the Chiso executives likely had very little visual knowledge of how morbid the effects of mercury poisoning are, making it easier to disregard any concerns and continue production. Now, in the context of gene editing technology, delayed or multi-generational effects, plausible deniability, and morbidity, we look at the invisible damage Minamata disease inflicted on pregnant women. In 1961, the pollution had still not ceased even after wastewater treatment efforts and government surveys were conducted, which were likely deliberately ineffective and just for show because they never released the results. So they started to notice a suspicious amount of cerebral palsy cases in newborn babies mirroring the symptoms of those with Minamata disease, with rather grotesque deformities. These babies were born after the initial outbreak, never consumed any seafood or shellfish, and their mothers never showed any symptoms of Minamata disease. How could this be? Understandably so, the mothers absolutely refuted the claim that their children had been affected by the disease. However, it turns out that methylmercury, when ingested by pregnant women, is removed from the mother's bloodstream and absorbed by the fetus, meaning that all the contaminated food the mothers presumed wasn't contaminated due to the absence of symptoms was being funneled right into their unborn child. Minamata disease had successfully permeated itself through people's genes under the radar and mutated a small generation of children. For current gene editing technologies like germline editing and CRISPR, where the changes made to one's genes are inheritable, we can use the case of Minamata disease in pregnant women as an example of an undetected mutation. So now I'm going to introduce the concept of cumulative error, or drift. This refers to the accumulation of error after an initial small error is made, gradually offsetting more and more as the process continues. It does not matter how small this error is, it will always increase. For example, a clock that is out of sync by just 1 second will eventually and gradually offset to far more than 1 second over time. This is a fundamental concept in mathematics and science, which happens to also apply to gene editing technologies like CRISPR-Cas9. Genomic dynamics are incredibly complex, with a high chance of generational load from off-target germline edits. A single off-target mutation in a gene editing experiment may help initially, but if it occurs in a regulatory region, it could subtly alter gene expression. From a purely mathematical or theoretical lens, over time, the cumulative effect of these edits can be described as linear or exponential divergence, depending on whether the errors compound. For example, if an off-target edit disrupts DNA repair mechanisms, increasing the likelihood of further errors. Assuming the errors do compound in one case, nonlinear cell division leads to cascading entropic mutations of dehumanizing character, accelerating DNA into synthetic and eugenic singularity meltdown in the most extreme cases among many other unforeseen consequences. but that probably won't happen, right? As it turns out, we have a chance in real life to see what actually happens when genetically modified humans, uh, you know, grow up or perhaps have children. Because in 2018, a Chinese scientist edited the genomes of 2 human embryos to resist HIV. The scientific world, they all basically condemned him, not just for operating without a medical license and violating laws, but out of the fear of the accelerated and multi-generational effects that could take place down the line in the HIV-resistant human's future lineage, making the development monumental but incredibly reckless. So, aside from this real-life scenario, we can also just think of some potential scenarios where CRISPR-based technology can take a turn for the worse. For example, in a mosquito population engineered with a CRISPR-based gene drive to reduce malaria transmission, an off-target mutation might reduce fitness in some individuals. Over time, this could lead to a population with unintended genetic diversity, potentially undermining the gene drive's purpose. Another example that kind of expounds upon the clock analogy in cumulative error. In this case, it's called error propagation. They're basically the same thing, but CRISPR experiments often involve iterative rounds of editing, especially in complex applications like creating disease-resistant crops or developing therapies for genetic disorders. Each round of editing introduces a risk of new off-target effects or other errors, where some cells have the edit and others don't. Each round of editing is like a tick of the clock, where a small error, off-target edit, or incomplete repair adds to the cumulative offset. Over multiple rounds, the genome may diverge significantly from the intended design, potentially leading to reduced efficacy or unforeseen consequences. In developing a CRISPR-based therapy for sickle cell disease, iterative edits to optimize the therapy might introduce minor off-target mutations. If these accumulate in the edited stem cells, the long-term safety of the therapy could be compromised. So there's also an epigenetic and functional factor to gene editing technologies. CRISPR edits can sometimes cause subtle changes in gene regulation or epigenetic markers, for example, DNA methylation patterns, and they aren't immediately apparent. These changes can lead to functional drift, where the edited cells or organisms gradually exhibit altered behaviors or traits over time. For example, an edit intended to knock out a disease-causing gene might inadvertently affect regulatory elements. Over time, this could lead to altered gene expression patterns, which might only manifest as a phenotype after many cell divisions or in specific environmental conditions. So, the ethical implications of gene editing technologies like CRISPR are, to be honest, no one can really say what they could specifically be, besides the fact that whoever decides to have their genome edited, which no one can decide that, but whoever decides to have their child's genome edited, they risk the multi-generational effects, but no one really knows what they are. That is why they're called unforeseen consequences. So, if we can't really predict what's going to happen, at the very least we can do is generate a set of guidelines to adhere to. So, 1. Improved specificity. High-fidelity CRISPR variants like Cas9HF or base editors reduce off-target effects, minimizing the initial offset. 2. Screening and validation. Next-generation sequencing is used to detect off-target edits after editing, allowing researchers to select cells or organisms with minimal errors. 3. Population monitoring. In gene drive applications, continuous monitoring of edited populations helps detect and mitigate unintended genetic drift. 4. Reversibility. Some CRISPR systems are designed with kill switches or reversible edits to limit the spread of errors over time. So, in the context of CRISPR, the concept of drift manifests as the accumulation of off-target edits, unintended genetic changes, functional deviations over time, cell divisions, or generations. Terms like off-target drift, genetic drift, or error propagation are all prevalent in different ways depending on the specific application. These effects highlight the importance of precision in CRISPR design and long-term monitoring to prevent significant divergence from the intended genetic outcome. Seeing what happened in Minamata with the fact that these invisible effects happen even outside of complex gene editing, if gene editing is to be continued in humans, it's imperative that we don't let it sneak up on us. That's all I've got. Thank you for listening.